Welcome to the Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Before we dive into today's topic, please take a moment to like this episode and subscribe to our channel. It really helps support the Science Experience and allows us to keep bringing you these deep dives into cutting-edge research. Today we're going deep into a topic that, well, it touches everyone: aging. Think about it. Back in 2019, maybe 1 in 11 people globally were 65 or older. By 2050, that number, it's projected to jump to 1 in 6. Huge shift. And this isn't just numbers on a page. It's creating this really urgent need for new kinds of therapies. We're not just talking about adding years to life, but really improving the quality of those years. Preventing or treating all those diseases that often come with getting older. Our guide for this discussion is a really interesting preproof that just came out. From Clock to Clock: Therapeutic Target Discovery for Aging and Age-Related Diseases by Chen and colleagues. So our mission today is to unpack how this revolutionary approach using aging clocks together with AI is basically changing the game in the hunt for what they call dual-purpose drugs. These are medicines designed to hit both the underlying aging process and specific illnesses at the same time. So, okay, how are scientists actually doing this? Let's get into it.
Well, what's truly revolutionary, I think, is the potential here to save, incredible amounts of time and money. Traditionally, finding a new drug it's a marathon. Years, sometimes decades, and hundreds of millions of dollars, easily. The idea behind dual-purpose therapeutics is actually quite elegant. You design a drug to target a specific disease, yes, but also the broader aging processes that, feed into that disease, and often others too. The advantages are pretty clear. Potentially shorter development times, lower costs, because one drug could tackle multiple issues. And maybe some of the early testing could even be streamlined. Plus, if your first target indication doesn't quite pan out, maybe you can pivot. It's about building in more efficiency right from the start.
That ability to pivot, that efficiency, it feels like a massive shift. And it's interesting, we sort of already have examples of drugs that accidentally do this, right?
Yeah.
Even before we had this targeted strategy.
Yeah.
Like Rapamycin.
Exactly. Rapamycin is a classic case. It comes from a soil bacterium, Streptomyces hygroscopicus. Its main job is inhibiting something called mTOR kinase. Now, mTOR is like a central controller for cell growth, metabolism, immune function, and, aging.
Okay, so it's pretty fundamental.
Very. It got FDA approval way back in '99 for preventing organ rejection, but since then its uses have expanded. And here's the dual purpose part. It consistently extends lifespan in, all sorts of lab animals: yeast, worms, flies, mice. And there's even human evidence suggesting it can improve some age-related immune parameters, like boosting the response to a flu shot in older adults.
That's pretty compelling. But it's not quite that simple, is it?
No, definitely not. There's important nuance. Rapamycin's effects are complex, often very dose-dependent. For instance, it can actually hinder muscle growth in young, active people. But in older animals, it sometimes helps preserve muscle mass, fighting off sarcopenia. The big challenge for using it more broadly against aging is the side effects seen at those higher therapeutic doses, plus some mixed results in studies. So it's not a magic bullet you just hand out.
Right, context matters. And then there's Metformin, another really common one. Huge for type 2 diabetes.
Yeah, metformin's fascinating. It has roots in traditional European herbal medicine, believe it or not, from goat's rue. Now it's the go-to drug for type 2 diabetes worldwide. Its mechanism involves activating AMPK and tweaking other pathways like SIRT-T1 and mTOR, all linked to aging.
So similar pathways to rapamycin in some ways.
Overlapping, definitely. And while it's mainly for diabetes, it's sometimes used for PCOS, though that's debated a bit. What's more interesting, perhaps, are these large observational studies in diabetics showing metformin use is linked to lower risks of cancer and heart disease.
And like rapamycin, lifespan extension in animals.
Yep, worms, flies, rodents again. And there's preliminary human data from big data sets like the UK Biobank and trials like MILES hinting at anti-aging effects.
But again, I'm guessing there are caveats for people just thinking about taking it for longevity.
Absolutely, especially for people who don't have type 2 diabetes. It can, for example, potentially blunt some of the beneficial effects of exercise on mitochondria and muscle growth. It's also been linked to lower testosterone in some men and, with long-term use, vitamin B12 deficiency.
Okay.
So, lots of promise, but also things to watch out for, particularly if you're otherwise healthy. It really shows why we need more targeted research, finding the right dose for the right person.
Okay, so these examples show the potential, but they were kind of accidental discoveries in a way. How do we move beyond lack? How do we design these dual-purpose drugs systematically?
That's where understanding the fundamental biology of aging becomes critical. You mentioned the hallmarks of aging. That framework is key. These dual-purpose targets we're looking for are essentially genes or proteins that sit at crossroads. They're involved in multiple processes that go wrong in both aging and specific age-related diseases. They're convergence points.
And finding those points, that sounds like looking for a needle in a haystack.
It used to be, but this is where AI comes in and really changes things. Tools like PandaOmics can sift through massive biological data sets. They combine that data analysis with the Hallmarks framework to flag the most promising potential dual-purpose targets.
Can you give an example?
Sure. Take TNIK, that stands for TRAF2 and NES interacting kinase. Originally, scientists knew it played a role in the cell's structure, the cytoskeleton, but now it's linked to cancer, metabolic issues, even neurological problems. And when you assess it against the hallmarks,
How many does it hit?
it's associated with 6 different hallmarks of aging. Things like chronic inflammation, how cells sense nutrients, cellular senescence, those zombie cells, even genomic instability.
Wow, 6.
Yeah, it's hitting multiple core aging pathways. That's why it's exciting. And in fact, it's already being tested in clinical trials for idiopathic pulmonary fibrosis, or IPF, which is a nasty age-related lung disease.
Okay, 6 is impressive. Are there others that hit even more?
There are. Another great example is Apelin. It's a natural molecule in our bodies that signals through the APLNR receptor. And Apelin, well, it's associated with 8 hallmarks.
8? Seriously?
8. It influences stem cell exhaustion, reduces inflammation, improves nutrient sensing, helps with cellular cleanup, affects telomere shortening, reduces senescence. The list goes on.
It's incredible. It's like a master regulator.
It's certainly involved in a lot of crucial processes. And like TNIK, it's already being investigated clinically for things like diabetes, pulmonary hypertension, and heart disease. So connecting this back, these targets like TNI and Apelin are so promising because they don't just tweak one thing. They seem to influence multiple fundamental mechanisms of aging all at once.
And you mentioned AI helps find these.
Yeah.
How much faster are we talking?
Oh, dramatically faster. We're seeing tools like BioBERT mining scientific literature, AlphaFold predicting protein shapes, even AI designing new drug molecules.
And that TNIK example you mentioned,
Yeah.
that was AI-driven.
Yes. The INS 018055 case study from In Silico Medicine is really remarkable. They used PandaOmics to identify TNIK for IPF, then used another AI platform, Chemistry 42, to design the inhibitor drug. The whole process, from picking the target to having a drug ready for preclinical tests, took just 18 months.
18 months compared
to the traditional 5, 10, maybe more years. And the cost was under $3 million versus potentially hundreds of millions.
That is unbelievable.
It really is. And that drug is now in phase 1 and phase 2A trials, showing it's safe and seems to be working as expected. It's not just faster and cheaper, it's a smarter, more targeted way to develop medicines. It feels like a paradigm shift.
Okay, so AI helps find targets like TNIK and Apelin by looking at pathways and hallmarks. But how do we measure if a drug is actually, slowing down aging itself? That brings us to the aging clocks, right?
Exactly. Aging clocks are the measurement tool here. They aren't physical clocks, of course. They're sophisticated mathematical models, usually built with machine learning. They're trained on huge data sets of biological markers, often DNA methylation, which are chemical tags on your DNA or maybe gene expression levels.
And they predict, what, just your age?
They can predict your chronological age pretty well, but more importantly, they can estimate your biological age, your risk of mortality, or how fast your body is functionally declining.
And how do they help find new drug targets?
By looking inside the model, essentially. The algorithms figure out which biological features specific genes, or those DNA methylation sites called CpG sites, are most important for predicting age or health outcomes. Those important features often point directly to potential therapeutic targets.
Okay, so there are different kinds of clocks using different data.
Right. The most well-known are probably epigenetic clocks. They look at those stable DNA methylation patterns. Think of them as tracking the body's long-term wear and tear. You've got Horvath's clock, Hannum's clock, and newer ones like Grimmage for mortality risk, or Dunedin pace, which measures the speed of your aging. They're quite reliable, though there's always some variability.
And then there are others.
Then you have transcriptomic clocks. These look at gene expression, which genes are turned on or off. They're more dynamic, like a real-time snapshot of your aging process right now. They give different information, maybe more immediate, but they can fluctuate more and tend to be costlier to run.
So one gives history, one gives a current snapshot. can you combine them?
Absolutely. That's the idea behind multi-omics clocks. They integrate data from epigenetics, transcriptomics, proteomics, you name it, for a much richer picture. And some new models, like Precious 1 GPT and Precious 3 GPT, are specifically designed not just to measure age, but to identify these dual-purpose therapeutic targets by linking aging patterns directly to disease risk within the model itself.
Wow. And is there evidence these clocks are actually pointing to useful targets?
Yes, quite strong evidence, actually. A big analysis looked at genes strongly associated with various aging clocks. They found that, on average, 27% of those clock-linked genes were already known targets for existing drugs.
Okay, 27%. How does that compare?
That's significantly higher than the baseline across the entire druggable genome, which is around 16.9%. So the clocks seem to be preferentially highlighting biologically relevant druggable pathways.
And does this connect back to TNIK and Apelin?
It does. Both TNIK and Apelin, which came up through the Hallmarks and AI approach, were also independently flagged as important by multiple different aging clock studies. This convergence is really powerful, different methods pointing to the same targets.
So the clocks aren't just for finding targets, they have a role later on too, in testing the drugs.
Definitely. They're incredibly useful throughout the whole process, especially in clinical trials. One way is population stratification. Clocks can help identify people who are aging faster or slower biologically, allowing researchers to select more specific groups for trials. Makes the results potentially clearer faster.
But you need to be careful which clock you use.
Right. Using just one clock might introduce bias, so the recommendation is often to use a panel of different clocks for a more robust assessment. They're also great for disease and treatment monitoring. We often see that people with age-related diseases have a biological age that's older than their chronological age.
Like an early warning system?
Sort of, yeah. And during a trial, the clocks can give a quantitative measure. Is this treatment actually slowing down aging at a molecular level? Is it reversing some age-related changes? It's a way to see effects beyond just clinical symptoms.
Are trials actually using them like this now?
Yes, increasingly. We saw the TRIME trial report epigenetic age reversal with a hormone cocktail. The calorie study showed calorie restriction affected glycan age. Senolytics trials are looking at DNA methylation changes.
So it's happening. But are there hurdles?
Oh, for sure. Cost is still a big one, especially for large trials. The TAME trial, which is testing metformin for anti-aging in thousands of people, apparently considered using epigenetic clocks, but found the sequencing cost prohibitive for that scale.
That's frustrating. The tool exists, but it's too expensive to deploy widely yet.
Exactly. But things are moving fast. You have initiatives like the Experize Health Span competition pushing for better, cheaper biomarkers, including clocks that can show effects within a year. And consortia are pushing for better standards, more data sharing, transparency, diversity in studies, and really focusing on using multiple explainable clocks so we understand why the clock is changing.
Explainable clocks, meaning you know which biological process is driving the change.
Precisely. It's not just a black box number. You can link it back to inflammation or senescence or metabolism.
So pulling this all together, there's this idea of a clock-to-clock framework. What's that about?
It's a proposed strategy, kind of a roadmap. You start with an aging clock to help identify a new anti-aging target. You then validate that target using the hallmarks of aging assessment, develop your drug. Then, in the clinical trials, you use a panel of diverse aging clocks to measure the drug's effect at the molecular level.
Okay.
And here's a clever bit. If the clock you use to find the target is also in your trial panel, you might use a modified version of that clock one, where the target gene itself is removed from the population to avoid circular reasoning or bias.
That makes sense. Now, what about the practical side? We talked about Rapamycin and Metformin being mostly generic, hard to fund big trials. How does this new framework address that?
That's a crucial point. The clock-to-clock approach is really geared towards developing new drugs like the TNIK inhibitor INS018055. Because these are novel molecules, they'll have patent protection. The goal is to have enough patent life left after the initial trials, say 10 or 12 years, to make it commercially viable for companies to invest in expanding the drug's use for multiple age-related indications.
So the AI-driven speed helps preserve patent life for broader application later.
Exactly. It creates the runway needed to explore that dual-purpose potential fully. INS018055 is kind of the poster child for this new paradigm.
It really feels like these different streams, the deep understanding of aging biology, the power of AI, and these precise aging clocks are all converging now.
They absolutely are. It's creating this incredibly exciting moment where we can realistically aim for these innovative dual-purpose treatments.
Treatments that could genuinely help tackle the huge healthcare challenge posed by our aging global population, improving not just lifespan, but health span. So here's something to think about. If we can accurately measure our biological age and actually develop drugs that treat diseases and potentially turn back that biological clock, what does that really mean for how we think about health, aging, and longevity in the years to come? Thank you for joining us on this deep dive. Remember to like this video and subscribe to stay informed. It really helps support the Sand Science experience.